Tumors of Central Nervous System, CNS. So the question is why we are going to deal about this tumor of CNS. Okay. So as you know, the tumor of the CNS is very high yield chapter for the examination point of view. So we can't neglect this chapter, okay? And it's not the content is too much. That's why we bother to go through this chapter, because this topic is quite boring. But I will try to make it as simple as possible, okay? So if you talk about the tumors of CNS, then if you know the structures of the central nervous system, then definitely you can know the data structures are going to have the tumor. That's why it's the tumor of the CNS. So let's get started. So before starting, you should just let's recall about the structures of the CNS, that is central nervous system. So in the brain or the nervous systems, we can encounter a lot of cells like the brain cells like brain parenchymal cells, like neurons, okay, the supporting cells, glial cells, okay, and covering, okay, knob coverings like the knob sheath, okay, knob sheath, and even the brain parenchymal covering, that is meninges, meninges. So these are the tumors which are associated with, or we can encounter in case of the central nervous system tumors, right? So let's get started. So this is the classification. We are going to make it in a simplified way as far as possible, okay? So the first tumor you can get here is, let's say, glial cells tumor or a glial tumor. These are the supporting cells of the brain, glial cells. So the next you can see here is poorly differentiated tumors, okay? So the third one is, we can say, meningeal. So meningeal tumor, I'm writing just meningeal, okay? So the fourth one you can write here is neuronal, okay? So here you can write the fifth one. So fifth one is nerve sheath tumors, and the sixth one is others, okay, other tumors or others. So we'll write all the tumors falling under this broad topic, okay? Okay, so under the glial tumors we can encounter, the glial cells are astrocytes, okay? So it will be astrocytoma. So the B1 will be ependymoma. So C is oligo. This is also a glial tumor, supporting cells of the brain, okay? So glioma, okay? So under the poorly differentiated, we can see, very important, medulloblastoma, right? Then neuroblastoma. So what you can just generalize here is all the blastoma are coming under the very poorly differentiated, okay? Neuroblastoma. We'll be talking in a detail about all these tumors, okay, in the upcoming session, okay? So in the meninges, definitely, if the tumor of the meninges, then it is going to be meningioma, okay, guys. So in the neuronal tumors, we can write neurocytoma, leave it only one, write only one here, okay, guys, because it will be just messy. We don't want to make these things messier, okay? We are trying to make it as simple as possible, okay? So next is knob sheath tumors. Definitely it will be schwannoma, okay, guys? So I'm writing here because there is no space. That's why I'm writing here schwannoma, knob sheath tumor, schwannoma, tumor of Schwann cells, okay? Because knob sheath cells are formed by cells, that is Schwann cells are knob sheath cells forming cell, okay? So others we can here write is, in others we will basically go to germ cell tumor, okay? T for tumor, germ cell tumor, okay, guys? And we can even encounter here in others lymphoma, okay, guys? So far we have discussed about the classification of the central nervous system, okay? Apart from this, the examiner will ask schwannoma, all these stuffs, all these contents mentioned here, any of them they can pick, okay, and ask in examination. So the latest examination question is the schwannoma. So right now I'm going to, in this very session, we are going to talk about or discuss about the schwannoma. and then followed by in a subsequent session we'll be discussing about all these tumors one by one in a very simplified way. Okay, so let's talk about the schwannoma first of all, because this is a very essential question. Schwannoma, schwannoma. Okay guys, so schwannoma, so how will be the question, what are the type of the question that they can ask from the schwannoma apart from the classification of the tumor of the central nervous system. Okay, so as you know, this swan cells, okay, this is a tumor of the swan cell, nerve cell tumor, right? So first of all, we will have to write a definition. Is this how this is occurring, okay, or where it has been arises, or how it is arising. Okay, so basically it arises from what? Neural crest, right? Yes, because swan cells are the derivative of neural crest cells. Okay, so schwannoma definitely, because it is the tumor of swans, that's why it arises from the neural crest derived swan cells. Okay, right guys, this is very important point. Okay, neural crest cell. So the second point we can write here is how this is occurring then. Okay, so it is basically associated with very important high point, eh guys, associated with NF2 neurofibrillary 2 type 2 mutation of chromosome, or which is present in the chromosome 22. Okay, this is very important. What will be the site? First, let me mention some point about this. Schwannoma is a benign tumor, guys. Benign tumor. Okay, benign tumor. So it usually do not metastasis, right? This is a characteristic of the most malignant, okay, neoplasia tumors. Okay, so okay guys, so the mutation is in a NF2 mutation of chromosome 22. This is something, you know, in the chromosome 22, the gene NF2 that is coding for the neurofibrillary protein, okay, that is going under mutation. That's why the schwannoma is occurring here. Okay guys, so talking about its symptoms, so symptoms arises due to local compression. So guys, how the symptoms arises, you can just correlate these things. If this nerve if the some nerve swan cells are undergoing the mutation and ultimately causing the schwannoma, you know, or the tumor, that tumor either can affect that very nerve, or can you know, cause compression to the underlying or the behind surfaces, or the surrounding adjacent surfaces. Because let's suppose this is a nerve, and because of the tumor, it is growing in size, and that's why the other parenchymal adjacent cells can be compressed. That will be causing the symptoms, right? So local compression of the involved neuron or nerve, simply nerve, or adjacent structures, right guys? So far we have discussed about the swan cells introduction, right? Or we can also say that about its ectopathogenesis. Okay, so the very important thing we must have to write in each and every cases of the tumors is the gross and microscopic feature. Okay, so let's talk about its gross feature. Okay, so gross feature. So as you know, this tumor is benign, means it is not metastasizing, or it is not invading the other, you know, other structures in a very deteriorating manner, right? And it is not undergoing the metastasis. That's why it is well circumscribed. Okay guys, encapsulated. This is most usual property of the benign tumors. Encapsulated, and usually if you don't you know, recall the color or the type of the tumors, you know, the type of the color of the tumors, then just write the gray color, because usually tumors show gray color or gray consistency. Also that's why I'm writing as a from gray marshes. Okay, so this is our first point. Okay, so second point will be it is attached to nerve, but can be separated. Yes, but can be separated, guys. Okay, but can be separated. The third point will be very important, very high point. Okay, So present with, or it is present with cystic and xanthomatous change. Xanthomatous change. Okay, guys. So just write 3 points about the gross feature of the schwannoma. Then very important high yield point, microscopic feature. Microscopic structure. Okay. So microscopically it shows mixture of two growth patterns. Okay, guys. So far we have discussed about introduction of the Schwann cell or Schwann cell's tumor. Okay, or that is schwannoma, followed by that gross anatomy. Gross feature of the schwannoma, and ultimately we are talking about the microscopic feature of the schwannoma. Okay, guys. So let's get started. So the first point we must have to learn is it may show the two pattern of the growth. That is, first one is Antony, a very very high yield point, guys. Antony A pattern, that is also known as cellular. Okay, cellular layer. Why it is called cellular layer? Because there are, more numbers of the tumor cells present in this very Antony pattern region. Okay, in this region. So, and what are the types of the cells? The cells usually we can encounter here, the tumor cells are usually elongated. Okay, guys, elongated cells with what? With cytoplasmic processes. In the area of high cellularity. What, guys? High cellularity. Okay. So in an Antony A pattern, cellular growth pattern, that is cellular layer. Why it is called cellular layer? Because this layer is usually composed of numerous amount of cells, right? Elongated cells. What types of cells? Elongated types of cells with the cytoplasmic process in the area of high cellularity, and that area where there is the high cellularity and these cytoplasmic processes are extending are something called as Vertical body. Very very high yield point. Okay, so the zone of high cellularity where these cytoplasmic processes present are called, guys, here I am writing because there is no space, are called Vertical bodies. So guys, this is very very important, must have to learn. Okay, so the second point I'm writing here, guys, we don't have a space, that's why I'm writing here the second point, that is Antony B pattern. Definitely this will be hypocellular because that is cellular, so hypocellular. So here you can encounter in Antony B pattern hypocellular tumors with a loose meshwork of the cells, microcyst and myxoid stroma. Hypocellular tumors with loose meshwork of cells, okay, microcyst and myxoid stroma, guys. Okay, guys, so far we have discussed about the tumors of the CNS. That is, if the examiner may ask the question in the way like we can see here, that is, classify the tumor of CNS and then list the most common site. Okay, guys, here I am just missing one very very important point. Let's mention here the site of the schwannoma. Most commonly it is found in the inferior vestibulocochlear. Okay, inferior vestibulocochlear nerve of cranial nerve 8. Okay, guys, 8. Very very ideal point. I just forgot to mention about that. Okay, so that is must learn this. So how the question will be? The question will be classify the tumors of the CNS central nervous system, and we can write these things, okay, one by one. Then list the most common site of the schwannoma. So the most common site is the inferior vestibulocochlear nerve of the 8 cranial nerve. Okay, so where it arises from the nucleus cells, drives Schwann cells. Okay, and how it is occurring, how it is happening, what is A2 pathogenesis? It is associated with the neurofibrillary 2 mutation gene of the chromosome 22. Okay, So the symptoms arises due to the local compression of the involved nerves or the adjacent structures. Yeah. So far we discussed about the schwannoma introductions as well as the classification of the central nervous system. Okay. Then we are going to talk about the we have even talked about the gross and microscopic feature of the schwannoma. So the most important point we must have to learn in a microscopic feature is the entering A pattern, that is also known as cellular, and the verrucate body is also encountered in case of the entering A pattern. Okay, and the B will be definitely the hypocellular. Okay, thank you guys.